And so was the monohybrid cross. The monohybrid is the result of the monohybrid cross. Now the taste cross. Taste to taste. What type of genotype? The dominant genotype. We want to know the dominant genotype. Showing a dominant phenotype and whose genotype is to be determined. So we want to know the genotype of the dominant phenotype as a result in F1, right? For example, whether a tall plant from F1 or F2 has capital D or small t capital D composition cannot be predicted. Therefore, to determine the genotype of a tall plant and F2, Mendel crossed the tall plant from F2 with a dwarf plant. This is called a test cross. Okay. So results of typical test cross where violet color flower into capital W is dominant over a white color flower, small w. So this is the representation of a test cross where we want to know the dominant phenotype which has genotype unknown. Can be capital W, small w. So we cross it with homozygous recessive. In here also homozygous recessive. So when all flowers are violet, it means the dominant phenotype was homozygous. If the all flowers are violet, you see no recessive in there. So if it is heterozygous dominant phenotype, capital W, so small w can be seen in the resultant, can be white. So half of the flowers can be white. So unknown flower is heterozygous. In here unknown flower is homozygous dominant. In here unknown flower is heterozygous dominant. Okay. So this is how we predict the zygosity of dominant phenotype, the genotype. See, so this is it. This is a test cross.